Toxicology, pharmaceuticals and fine chemicals. Introduction to the module. Toxicology is a scientific principle that examines the potentially negative or harmful effects of subjective chemicals on living organisms. It combines principles from chemistry, biology, and medicine. This module explores the uses and concepts of toxicology whilst acknowledging its links to pharmaceuticals and other chemical usages. Topics to be covered: toxicology, sources of toxic compounds, movement of toxicants in the environment, pharmaceutical fine chemicals, pharmaceutical active ingredients, peptides, nucleotides and saccharides. Toxicology. Toxicology can be simply defined as the study of poison, which can be any substance that can potentially cause harm to a living organism. However, this simplistic definition can have significantly broader meanings when it comes to the classification of poison and the measurement of toxic effects. The practice of toxicology can include detection, investigation, verification, reportage, and the regulation of toxic material. Those actions may not fully resolve all outstanding issues, and an incident of toxicity is more than just a one-off exposure to poison. Exposure will be invariably followed by distribution, metabolism, cellular changes, and a toxic endpoint expression. Poison is often a quantitative concept, as almost many substances can be harmful in large doses, but harmless in smaller quantities. There can be a range of impacts, which can run from subtle long-term chronic toxicity to immediate lethality. Vinyl chloride may be taken as an example. Toxicity may be acute or chronic and can vary in its effects from one organ to another. Age, genetics, gender, diet, physiological condition, or an organism's health status can also have an impact on a toxin's symptoms. Since humanity has extreme genetic diversity and variation, this is an important part of toxicology, whereas animals have little genetic variations in similar species. Clinical drugs are especially diverse examples of toxins, as most are therapeutic and highly beneficial at some doses. However, higher doses of certain compounds have drastic effects on the human body and can easily result in long-term damage and fatalities. This is what makes the measurement of toxicity in a chemical so complex. Aspirin is a relatively safe drug at recommended doses and is taken by millions of people worldwide. At the same time, chronic use can cause harmful effects on the gastric systems, and it is fatal at a dose of about 0.2 to 0.5 g per kg. Approximately 15% of reported accidental deaths from poisoning in children result from pain relief medication, including aspirin. Sources of toxic compounds. It is not easy to classify a chemical compound as being inherently toxic either by chemically or practical means. Some are natural products, many are synthetic organic chemicals used for legitimate medical purposes, and some are byproducts of industrial processes and waste disposal. The categories of toxic compounds are: exposure classes. Exposure classes include toxicants in food, air, water, soil, and toxicants characteristic of domestic and occupational settings. Use classes. Use classes include drugs of abuse, therapeutic drugs, agricultural chemicals, food additives and contaminants, metals, solvents, combustion products, cosmetics, and toxins. Some of these, such as combustion products, are the products of use processes rather than classes. Movement of toxicants in the environment. Chemicals released into the environment rarely remain in their original form or stay at one location. For example, agricultural chemicals used as sprays may drift from the point of application as air contaminants or enter runoff water as water contaminants. Many of these chemicals are susceptible to fungal or bacterial degradation. They are rapidly detoxified, frequently being broken down into products that can enter the carbon, nitrogen, and oxygen cycles. Other agricultural chemicals, particularly halogenated organic compounds, are resistant to metabolism by microorganisms and persist in soil and water as contaminants. They may enter biological food chains and move to higher trophic levels or persist in processed crops as food contaminants.